regarding the membrane, remember that the plasma membrane, the whole foundation of the plasma membrane are phospholipids, or is phospholipids. And so that means that any lipid-soluble substance, another way to say that is any hydrophobic substance, or water-insoluble, non-polar, say this several different ways, but anything that is basically a lipid or a lipid-soluble doesn't have to have any special mechanism to get in and out of the cell. Anything that is lipid-soluble, hydrophobic, non-polar, water-insoluble
Just goes through.
just slides right between the phospholipids.
So it's passive.
Yes. Okay. Now, what's really cool about this is you wouldn't think about it, but oxygen and carbon dioxide are lipid-soluble. They don't dissolve very well in water, but they dissolve excellently. They are very soluble in lipids, which is very convenient because that means that oxygen can slide right in and carbon dioxide can slide right out. However, most of the stuff in your body is water-soluble or polar or hydrophilic. So these things cannot just slide in between the phospholipids. They've got to have some kind of protein, some kind of transport protein, either a carrier or a channel, in order to get in and out of the cell. The smallest water-soluble things, like ions, can use those channels, the tunnels. A little bit larger molecules, like amino acids and glucose, use the carriers. And if it's a really large molecule, then you have to use a vesicle. So things like proteins, very large substances, have to use a membranous sac or a vesicle. The birth control patches or the nicotine patches, how do they work? The stuff that's in that patch is lipid soluble. So it just slides right through the cells, right into your bloodstream. In all of this together, there are two basic ways to get stuff in and out of the cell. You either do it the free way or the expensive way. If the cell doesn't have to spend any money, the cell doesn't have to use any energy, we call that passive transport. If the cell has to use energy, if it's got to spend some money, if it's got to do some work to get stuff in or out of the cell, that's active transport. Now, your book does something that just drives me crazy. When I think about passive and active transport, the first thing I think of is diffusion. And you either have what's called simple diffusion or what's called facilitated. When you facilitate something, you're helping it. So you can have simple diffusion or you can have facilitated diffusion. And then you can have what's called osmosis. So if you're thinking about this, passive transport is basically diffusion or osmosis, and then you can have two types of diffusion. Does that make sense? And then under active transport, you have what we would call primary active transport, secondary active transport, and then vesicular transport. This is the way it is organized in my mind. But what happens is, your textbook does something funny. It throws a category called carrier-mediated. And what that is facilitated diffusion is a type of carrier-mediated. It means it uses a carrier protein. Facilitated diffusion is carrier-mediated, but it's passive. Primary and secondary active transport use a carrier protein, but they're active. What's the difference between diffusion and osmosis? Diffusion is stuff. Osmosis is water. Now, by stuff, I mean the solutes, right? So, molecules can diffuse through air, they can diffuse through water. All of these things, all these passive methods here. We say that the stuff moves down a concentration gradient. Now, what's a concentration gradient? It just means that there is a difference in concentration of some substance from here to here. There's a bunch of it over here. There's not a bunch of it over there. The difference is the concentration gradient. So stuff moves downhill. It moves from where you've got a bunch of it to where you don't have a bunch of it. And it does this because the molecules are randomly moving. And they're bumping into each other, and they eventually just bump into each other enough that they're spread out evenly. So we say diffusion is the movement of a solute from an area of high concentration to an area of low concentration. Heat is basically the movement of the molecules. And unless you reach absolute zero, which means there's absolutely no heat whatsoever, molecules move. I mean, there's electrons zipping in and out of existence, and so you've got little bitty molecular movement. In fact, if you look at bacteria in a little bit of fluid under the microscope, they wiggle. Even bacteria that don't have ways to move, they wiggle because of Brown. It's called Brownian motion. Brown was the guy that discovered this. And it's just the water molecules bumping into the bacteria, basically. So all the little molecules in your body are moving. Yeah. But it's so small on a macroscopic scale you can't see it. But even if you use a light microscope, you can detect the movement. Okay. And this is passive. It doesn't require any energy because the energy is in the molecules themselves. Diffusion is fastest if you don't have to go very far, if the molecules are small, if the temperature is high, because the higher the temperature, the more the molecules are bouncing around. And so the molecules themselves have more energy. And if there's a big difference in concentration, if the gradient is large. Now, the other thing that can help diffusion is if you have different charges involved. So, for instance, if I have a cell, and for some reason the inside of my cell is negative and the outside of my cell is positive, then that means that anything that's positive wants to get sucked into the cell because the inside's negative. So all of these conditions help increase the rate of diffusion. Now, the good news is, across the cell membrane, basically, you've got 2 layers of phospholipids, right? That's a pretty small distance. Initially, you've got more stuff over here than over here. And so, these things diffuse over there. But at some point, it balances out. And so, initially, the net flow is in that direction. But once you no longer have a concentration gradient, the net flow decreases as they begin to balance out. Because what happens is, once they're balanced out, once they reach equilibrium, then every time a molecule diffuses in, another one diffuses out. And so that's why the net flow goes to 0. Once there is no longer a concentration gradient, net diffusion doesn't occur. Because there's nothing driving it. If you've got one molecule going in, another one comes right back out again. So there's no net change in concentration on either side of the membrane. Lipid-soluble substances undergo simple diffusion. Because they're lipid-soluble, they don't need a transport protein. They slide right through those phospholipids. But all the rest of this stuff over here, all of these water-soluble things, have to have some kind of protein. So water-soluble substances use facilitated diffusion. Facilitated means there's a protein needed. Water-soluble substances have to have a transport protein. They just can't cross that membrane by themselves. They don't dissolve in those phospholipid, the hydrophobic tails of the phospholipids. They can't just slide right through. Now, facilitated diffusion can use, and this is where I differ a little bit with the book. Facilitated diffusion, your book says facilitated diffusion only uses carriers. Facilitated diffusion, you can use a carrier or a channel, not a vesicle. For instance, let's say here's our cell. And let's say we've got a lot of sodium ions on the outside. Got a whole bunch of these sodiums out here. And we don't have much in here. Now, sodium, it's a charged molecule. It's an ion, right? Not a molecule. It's a charged substance. It's polar. It's water-soluble. So the only way that sodium can get in here, it wants to get in here, right? Because you've got all these sodium ions bouncing around out here, and they really would like to get in here where there's not any. But the only way they can get through is if there is a sodium channel. Channels and carriers are selective. So, if this cell has a sodium channel, then sodium can diffuse into the cell. It's diffusion because the sodium is moving from an area of high concentration to an area of low concentration, right? It's facilitated because you have to have a protein channel.
If the cell doesn't need that, would it matter that it has a high concentration and low concentration?
If the cell doesn't need it, what it does is the nucleus says, okay, quit making sodium channels. And so the sodium will stay outside.
Okay. Oh, so they don't just stay there? You don't keep your channels?
Oh, no. The cell can change the channels. It can change its receptors. The cell can change all of those proteins at any time whatsoever.
To get what it needs.
To get what it needs, to detect what it wants to detect, to ignore what it doesn't want to do. That cell membrane, that's a good point. That cell membrane is a dynamic. It is constantly changing. The proteins, they're embedded in those phospholipids, but they can move around. It's almost like the phospholipid. In fact, sometimes it's called a sea of phospholipids. The proteins bob around in a sea of phospholipids. They're not even fixed in their spot. They can move around. So channels only work for very small substances, ions, and water molecules. They can't do anything much bigger than that. So if you need something bigger, you have a carrier protein. Carrier proteins work like a mount or a revolving door or
like Pac-Man.
It's like, yeah. What happens with carrier proteins, in this example, let's say this is a glycine. That's an amino acid. Let's say the cell needs some glycine. And so it puts some glycine carrier proteins in its membrane. The nucleus says, we need glycine, make some glycine protein. Okay. And so the glycine carrier protein gets stuck in the cell membrane. And so there's a lot of glycine out here. So I've got more glycine on the outside than I have on the inside. So glycine wants to come in, right? It's all bouncing around, knocking into each other. So eventually what happens is the glycine molecule binds to the carrier protein. It's got a little spot on it that catches the glycine. When that glycine does that, when it binds to its little spot, that causes a conformational change, which means in biology something happens that's cool, but we don't understand it. But the protein changes shape. The carrier protein changes shape, and it closes the little opening to the outside, and it opens the opening to the inside. So that's what I mean. It's like a revolving door. If you've got a concentration gradient, lipid-soluble substances use simple diffusion. Simple means no protein involved. Water-soluble substances have to use facilitated diffusion. Either a channel protein or a carrier protein, depending on how big it is, right?
But they still don't need energy.
As long as you've got a concentration gradient, as long as there's a difference in concentration, exactly. As long as you're trying to get the substance to go, if you've got a bunch of it over here,
The direction it should go.
the direction it should go, right? It's got to go downhill. If you want to do it the other way, you got to do this. But as long as you're trying to move the substance with the concentration gradient. Okay, osmosis is the movement of water across a semi-permeable membrane, which is what this is, right? It's semi-permeable or selectively permeable, either way you want to say it. It controls what goes in and out. Now, repeat after me. Salt sucks.
Salt
Y'all are
sucks.
so good. Water wants to go where the stuff is. If the stuff can't move, for instance, let's say here's my cell again, and my cell doesn't have any sodium channels. I got a bunch of sodium out here, right? And my cell says, I don't need that sodium. But what happens is, there's water inside this cell, right? Intracellular fluid. And what happens is, that salt, the sodium chloride, salt, right? Okay. That will suck the water out of the cell. Water wants to go where the stuff is. If there's more stuff outside the cell, the water gets sucked out. Now, if there's more stuff in the cell, and I just use salt as an example, it could be any solute. If there's a whole bunch of stuff in a cell, and not much stuff out here, the stuff sucks the water in. Now, and eventually what will happen, because our cells don't have cell walls, if enough water goes in, what's going to happen to the cell? Yeah, it bursts. Water is small enough that it can actually squeeze between them. It's not real fast, but it happens. Okay. Now, if you want it to happen faster, that's when you put those water channels, those aquaporins in there, and that speeds up the osmosis. But water molecules are small enough that they can squeeze in between the phospholipids. That's an exception. And it's just because they're so small. So you can have water channels, aquaporins, that make this process a lot faster. But it will happen automatically. It just takes a little bit more time. Water is the only molecule, the only polar molecule that can cross the plasma membrane without a channel. Any nonpolar substance can. Yeah, any lipid-soluble, hydrophobic, nonpolar, any of those slide right through. Now, if you want to speed up the movement of water, you can put some water channels in there. But you don't have to have those to get the water to move. When water moves, that force of that water, it creates a pressure, it creates a force. We talked about it. If too much water goes into the cell ruptures, right?
Yeah.
Okay, so we call that osmotic pressure. So the movement of the water is osmotic pressure. The force that it generates is hydrostatic pressure. The force pressing, if we've got water running into this cell, then that water starts to push against the cell membrane, right? That fluid pressure is hydrostatic pressure. The movement of the water, the force of the water moving in is osmotic pressure. The force that it creates pressing against the membrane is hydrostatic pressure. Blood presses against the inside of your blood vessels, right? What do we call that? Blood pressure. That's an example of hydrostatic pressure. You can have hydrostatic pressure against any surface. It can be inside of the cell. It can be inside of the blood vessel. It can be inside of your stomach. So we have the intracellular fluid. We've got the blood. between. Anybody know what that's called? Well, anything outside, including the blood, is an extracellular fluid. But the fluid that's not inside the blood vessels and not inside the cell, it's called interstitial fluid. Now, you have lots of protein in the plasma of your blood. Lots and lots of protein. One of the things your liver does is it makes these proteins, all kind of good proteins. That protein, the presence of that protein, helps make sure that the right amount of water stays in the blood vessel. Because as long as you've got the right amount of protein, then you've got the right amount of fluid in your blood. The protein is like a particle. It's dissolved in the plasma. And so if there's a lot of protein in there, the water wants to be in there with it, right? Your cells have a lot of proteins on the inside. And so you have a lot of protein inside your blood. You have a lot of protein inside your cells. And that's one way the cells control where the water goes. Now, you do not have a lot of protein here. Now, if you go into liver failure, one of the things that will happen is all of a sudden you don't have the right amount of protein. Guess what happens to the water? If the water in your blood doesn't stay there, it gets into the tissues, causes edema. That exerts pressure onto your cells. If it's a nerve cell, the nerve cell says, ow. That's one of the things that generates the pain signals. It's uncomfortable, right? If the pressure gets large enough, it hurts. So, you can have hydrostatic pressure against the outside of a cell. If you take a cell and you put it in fluid, let's say like your blood plasma, and it's nice and happy. Same amount of stuff out here, same amount of stuff in here. There's no net movement of water. If a water molecule goes into the cell, one slides out. We say that the solution that the cell is in is isotonic. And so there's no net change in the movement of the water molecules. If one goes in, another one goes out. If one goes out, another one goes in. Okay. And so this tonicity, isotonic, the tonicity refers to where's the water going. If it's isotonic, it's not really going anywhere. If you put the cell in pure water, you got all this stuff. If this is pure water out here, there's nothing dissolved in the water, just water, right? And you still got all the same amount of stuff. In fact, you got proteins, all kind of stuff in the cell, right? So where's the water going to go?
In the cell.
Yeah, the water's going to go into the cell, right? Because there's more stuff in the cell, so the water goes into the cell. That solution is called hypotonic. The amount of solute is low. So if you put a cell in a hypotonic solution, so that means that there's more stuff in the cell. Water goes where the stuff is. There's more stuff in here than out here. If you're dehydrated and you go to the emergency room, what do they put in your veins? Saline. Yeah, they don't put pure water, right? All your red cells will go. They have to put an isotonic solution into your veins. They can't put pure water. That's hypotonic. You blow your cells up. If you're in the middle of the ocean, don't drink the seawater. They've got more stuff outside than you've got inside. Where's the water go? Out. Yeah. The stuff sucks the water out of the cells. That is called a hypertonic solution. All right, so all of that is passive. All of that is passive. The cell doesn't have to spend any energy to make that happen.